Okay, everybody has to press понятно or something like this in order to accept the fact that we are recording this and behave appropriately. So now we share the screen. And I will share with you my presentation. Things are not going smoothly because software isn't perfect. All right. So if everybody can hear me and can see the screen, this should be good. Okay. So now the class name, the class title is Mathematical Aspects of EEG and MEG-based Neuroimaging. And this class is actually an elective course, is intended for selected students who have the necessary mathematical background. Okay, so you do have to be aware of calculus, or maybe even some advanced calculus aspects. Linear algebra is absolutely a must, and probably some idea of neuroimaging techniques that have been used and that are present around, and some interest into those is actually a needed component to be successful in the class. Okay, so I also would urge you to think abstractly, to learn to think abstractly as much as you can, and you should possess some MATLAB and Python coding skills, not to a great extent, but at least not to be afraid of those two tools. Okay, so have some reading. We will be giving you the chapter names after each lecture that you need to cover, to go over. We will be covering more in some aspects than it is written in the book, but in some more mathematical aspects, books will give you more advanced and more detailed introduction to the thing that we are working on. The software, as already mentioned, will be Python 3, MNE Python, and sometimes MATLAB we will use. And we will have grading policy. We will have homeworks, which will contribute 25% of the grade. We will have participation. This is not quantified formally. This is basically something I leave to myself to boost up your grade if you behave well during the class, and in a sense that if you participate, if you ask questions, if you attend the lectures at the very least, you know, you can think of it as a gauge for myself not to give you bad grades. Okay, and the 50% of the grade will come from the final exam. Okay. So this is the formal introduction, and the class is taught by 3 people. The first person is me. I mean, I will give you the majority of lectures. Then Daria Kleeva is our teaching assistant, who will conduct seminar sessions with you. So you will practice in primarily Python and with MNE Python software. And the first little introduction on what kind of things you will be dealing with in terms of software you will have today. After the very first lecture, that we are very glad to have Maria Volodina participating in the class, who is a researcher at the Center for Bioelectric Interfaces. And Masha has a very significant background in biology. She actually graduated from Moscow State University, both with MS and PhD degrees. And so she will give you an introduction to the signals that we are measuring with EEG and MEG, to their physiological origin. Okay, so she will talk about this zoo of ion channels and other very complex stuff. But I hope some of this you already know, at least starting to learn in your neuroscience class. So this will come for you like a brief summary of what you have to be aware of, so that you approach processing this data in some conscious way. Okay, so I'll pass my word to Masha now, I think, and I'll stop sharing my screen and hope that she will share her screen.
Okay, I'll try to do it now. Moment. So can you see my screen?
Yes.
Okay.
Yes, we can.
Yes, we can see.
Yes, and
Alex.
also last thing, very last thing, I'll tell you.
Okay.
Please try to participate in the class as much as possible. Interrupt Masha. I think it's possible, right? To ask questions. Not like if she considers that the question requires too long of an answer, then she will postpone answering the question given some very brief explanation. It will create the dynamic nature for this class because the class is complex. Okay, so it's difficult. Okay. So, and it's normal for you to not understand the lecture from the first run. In order to increase your understanding, please do ask questions, okay?
Okay, let me start. Alex, thank you for introduction. My name is Maria Volodina, and I'm working in the Center for Bioelectric Interfaces, and I have a PhD degree in physiology, as Alex said. And here's on the screen is my personal email. If you want to ask something, or I don't know, say you didn't like my lecture, or I have stupid questions, you can write here. Not my official email on the HSE page, but this one. And today I will tell you about physiological basics of EEG and MEG. And again, we really hope that you know at least the most of the things I will tell you today about. As you already had your neuroscience course and probably passed the exams, so it will be just a short reminder about these processes, about these basics. And we will start today with the very basics of electrophysiology. We will talk about electrical signal in neurons, about resting membrane potential, action potential, and postsynaptic potentials. Then we will talk about synapses and neurotransmitters, and we'll have some short review of receptors, different kinds of receptors. Then talk about very basic principles of EEG and MEG, and just a couple of slides about the origin of brain rhythms. And I will say in advance, we will have 3 short quizzes during this lecture. It will be just several simple questions. And I think you can answer these questions right now, but it will be during the lecture, and you should be ready to type the answers in the chat. Don't be stressed about it. And we will start. I hope you will. Yeah, we will start from resting membrane potential. What is resting membrane potential? It is the difference of electrical charge inside the cell, in the cytoplasm, and extracellular space on the outside the cell. And every living cell, not only neurons or muscular cells or whatever, every living cell has this difference, has resting membrane potential. But today we will talk mostly about neurons, as we have this about neuroscience courses and interest here. And in average neuron, there is about minus 60 or minus 70 millivolt of resting membrane potential. So it means that inner membrane of the cell is negatively charged relatively to the extracellular space, to the outer membrane of the cell. Where it comes from? It comes from, I try to remove this. Yeah. It doesn't look very nice. As you can't see. Okay, I will remove it. Alex, if you know how to, can I just remove it? Okay, there's a different concentration of ions in the cytoplasm and in the outside. And you can see here on the bottom the approximate concentration of ions in the neuron. And the important thing that inside the cell, in the adult neuron, there is high concentration of potassium ions and low concentration of sodium and chloride ions. I will talk about it later, but in the immature brain, in embryonic cells, there is another situation with chloride ions. There is opposite concentrations, high concentration inside and low concentration outside. But in adult cell, we have this high concentration of potassium and low concentrations of sodium and chloride. Why is it so? It works this way because there is different permeability of the membrane to different ions in resting state and in any other state. Charged ions can't just go through the membrane. They need some special pores, some special channels. You can see there. And in resting state, it is mostly potassium ions that are open. So it is mostly potassium can just go through the membrane and it go from the cell, it go out of the cell because of the concentration gradient, and it result in decrease or decrease of positive charge inside the cell, and it results in this negative charge on the inner space and the inner membrane. And the second important channel that also contribute to this resting membrane potential, it is, I will move it now, it is sodium potassium pump, TFS. It brings 2 ions of potassium inside the cell and 3 ions of sodium outside the cell. So it is totally also result in 1 minus inside the cell. Yeah, because 2 ions here, 3 ions outside. Here minus. So we have what we have. We have minus 60 or minus 70 millivolt inside the cell. And yeah, and here's just a couple of, I don't know, what is it, physics, mathematics, Nernst equation and Goldman-Hodgkin-Katz equation. Probably you know that. This is how you can calculate this equilibrium electrical potential difference if you need to do that. This Nernst equation is for a very simple system when you just have one kind of ions and you know the concentration inside and outside. And then you can use this and you will know what will be this equilibrium potential. Here on the right, you can see these approximate values for system where you have just calcium or just sodium or just, just potassium or sodium or calcium or whatever. But in real system, you have at least several ions. The main ions that contribute for the resting potential and in the cellular process is potassium, sodium, and chloride. And if you know their concentrations and you know permeability of membrane for these ions, this pK, then you can calculate the summary, this total potential, membrane potential you will have. And the next important property of the neural cell and also muscular cell, but again, we are talking today about neurons. It is action potential. Action potential is acute depolarization of the cell membrane. Depolarization can be also used the word hyperpolarization, and don't be confused. In some cases, you can see hyperpolarization. In some cases, you see hyperpolarization. So you shouldn't be confused about all these, both of these words. And depolarization or hyperpolarization, it is decrease of negativity. Why it happens in neural cell? In the resting state, we have our resting membrane potential. We have discussed already about here minus 70 millivolt. But then in some reason, some stimulus comes. For example, some electrical stimulus, or maybe some ion channel is opening and permeability of membrane changed. And we have this slow depolarization phase when the inner membrane became less negative. And when it reached minus 55, again, in this case, in this picture, it can differ a little. But here, it reaches gate threshold, and voltage-dependent sodium channels are open, and a lot of positively charged sodium ions come inside the cell. And we can see this quick, this acute depolarization, acute decrease of the difference between the inner and outer space, and in reaches even positive values. So the inner membrane becomes more positively charged than outer membrane. But in some point, these voltage-dependent sodium channels are closed, and voltage-dependent potassium channels are open with some additional potassium channels, and this potassium come inside the cell, and also there is active work of sodium potassium pumps. We discussed it already, and we can see this repolarization phase when the membrane becomes negative. And there is also this phase of hyperpolarization, when the membrane is even more negative than in resting state. And in this point, these additional voltage-dependent potassium channels are closed, and then go back to the resting potential, to this minus 70 millivolt. And this is important period because this hyperpolarization period when the membrane is more negative than in resting state, because in this period of time, the cell is even less excitable than in normal resting state. There is also a short period of absolutely refractory, when the cell can't be excited. You can use any stimulus you want, but it can't be excited. But then you just need to use more strong stimulus to excite it somewhere here. Yes, it was about resting potential, action potential, and then this action potential can propagate along the axon and go to the next cell. But before we discuss this transmission from cell to cell, we will have short quiz. It will be just 3 simple questions, and you will have 2 minutes to answer them. I will start my timer when I switch the page. And the most complex thing you need to do, you should type the answers in the comment chat and don't press send. Wait until I will say you, now send. And then we will just look, do you really know all these things I talked just about? Okay, let's start. Are there some questions now? No? Okay. Then start. Remove it. Sorry. Mm-hmm. Please send your answers now to the chat, and I will look. Okay, yeah. Okay, so the first answer is potassium. Ali.
And why is Mikhail Sinkin not answering?
Don't know. Maybe some problems.
Maybe he's at the operation, or I don't know, at the surgery room. Okay.
Okay, the last one. Yeah, okay. The answer to the first question is potassium ions. There was. Yeah, sodium, potassium ion. Okay, we'll just come back to this. Yeah. Again, about the first question. In the resting state, we have high concentration of potassium inside the cell. And this potassium that has the main contribution to the resting membrane potential. But also, there are some permanently open potassium channels in resting state, but also there is sodium-potassium pump that brings 2 ions of potassium inside the cells and 3 ions of sodium outside the cell. So yeah, it brings both sodium and potassium ions, but totally it results in 1 minus inside the cell. There are some questions about it? Yeah, if not, then we'll go.
No, well understood.
One more answer or question? Yeah, this pump is also active in the resting state, but on this repolarization state, it becomes even more active. So actually, it is all the probability history. There is no. It doesn't work like this channel is absolutely closed and this is absolutely open. No, the most of these channels are open or closed during the resting state, and then during the action potential or during this repolarization phase, it becomes even more active or less active. Not absolutely. Nothing in physiology, in biology works yes or no. No, just probabilities. Okay, and now we'll talk a little about signal transmission. The action potential can go along the axon and then reach another cell. It can be neuron or muscular cell or some other cell. And this place of contact between neural cell and some other cell, some target cell, is called synapse. And there are 2 big types of synapses. The first one is chemical. The second one, here on the picture, is the first or the second, but it seems to me that historically this one is the first. This electrical synapse. And we'll start with electrical synapse. It is very close connection of 2 membranes. And they have some special protein complexes that form these pores between 2 membranes. And so the cytoplasm and ions just can go through these channels. And the main property of, and it called gap junction. And the main property of this kind of synapses is signal transmission through it is very rapid. And if you need to synchronize a lot of cells, to excite them really simultaneously, this is very good solution. And for example, it is very abundant. It works in pyramidal cells in the brain. But what's, you can say, wrong with it, but in other property, that it can transmit only action potential. So it's just, it works like long axon. Just you have not a lot of cells, but one long cell. And you can transmit the signal only for some distance, because the action potential becomes lower and lower and lower, and the amplitude of it becomes smaller when it goes through these gap junctions. And the second one is chemical synapse. It is more complex. I would say it is more interesting, but it's very subjective. And I would say it's more abundant in human CNS and human brain. How it works? It works in this way. When action potential reaches this axon terminal, it depolarizes the membrane, and it causes the opening of voltage-dependent, voltage-gated calcium channels. Then calcium goes inside the cell and triggers that synaptic release of neurotransmitter from the synaptic vesicles. And it also triggers a lot of other processes. Calcium is very important ion in all the processes in the cell. But here we're interested in this, in release of neurotransmitters. Neurotransmitters go to the synaptic cleft. This space is called synaptic cleft. And from here, they can bind to the postsynaptic receptors on the postsynaptic membrane. So these receptors on the target cell. Or they can bind to the outer receptors. Outer receptors are also really important. And why can we use it? Usually, it works like negative feedback. So, for example, we have some kind of excitatory neurotransmitter. We'll talk more about this later, but now just excitatory neurotransmitter that cause excitation of the next cell. But here on the presynaptic terminal, we can have inhibitory receptor. So this neurotransmitter at the same time excite the target cell and inhibit the presynaptic cell. And it stops the transmission of the signal. It is very important in biological system, this negative feedback, as overexcitation can be very toxic, can be harmful, and can just destroy the cells. We need to stop the transmission of the signal. There are also some situations when there is positive feedback through these outer receptors, but much more frequent is negative feedback. And now we'll talk about these receptors. I have said about excitatory, inhibitory receptors, but at first, what kinds of receptors there are? There are, again, two big classes of receptors, dependent on the mechanisms, how they work. There are ionotropic receptors and metabotropic receptors. And first, ionotropic receptors, or it can be called ligand-activated ion channels. It is just the channel that is open or closed, so it somewhere change its conformation, its shape, when the neurotransmitter binds to it. And it works like it's usually some protein complex, and when you have the bind it neurotransmitter, it can in some way change its configuration because of quite complex biochemical and biophysical processes, but we will talk about it now. And it can, for example, open the channel for positively charged ions. And this, depending on type of ions that can go through this channel, it can have excitatory or inhibitory effects. And there are some very selective channels. For example, the channel that is just for sodium. or not selective, for example, channels that works with all positively charged ions. And these kind of ions result in some effect on the target cell. And the second type of receptors is metabotropic. Oh yeah, and these ionotropic receptors work very quickly. They produce very quick response. It takes about tenths of microseconds from the binding of neurotransmitters to the opening of the channel, but at the same time, their effect lasts for quite short period of time. It's about milliseconds usually. And yeah, the second type is metabotropic receptors. It's more complex. The binding of neurotransmitter to this receptor cause quite usually quite a complex cascade of reactions inside the cell. So, for example, it can cause the phosphorylation of one protein. This protein will, I don't know, phosphorylate another one or doing some other transformation and changes in the whole system in the cell. And as a final result, it can also indirectly regulate some ion channel, or it can have another effect. In case of indirect regulating of ion channel, it also works like ionotropic receptor, and depending on kind of the ions that can go through this channel, it will be excitatory or inhibitory. Or it can, for example, increase the expression of some proteins in the cell, or cause apoptosis or whatever else. And these metabotropic receptors are slower, but their effects can last for longer time. And I have already used the words excitatory and inhibitory effect potentials. And this is about postsynaptic potential. Here on the left, you can see examples of excitatory and inhibitory synapse. And for example, excitatory synapse, here you can see the release of neurotransmitters that cause influx of sodium ions. What happened in the cell? We have already talked about action potential here in the picture. But if this depolarization doesn't reach the threshold level and action potential doesn't start, this is called just depolarization. And in the cell, in the target cell, it will be excitatory postsynaptic potential. If we can see this slow increase, decrease of negativity, increase of potential, and this picture is not very important because postsynaptic potential is longer than action potential. It should be like this. Yeah, and this is excitatory postsynaptic potential. Why it is excitatory? Because it makes the cell more excitable. We need we need less stimulus. We need weaker stimulus to start to induce action potential from this point. And inhibitory postsynaptic potential, it is hyperpolarization of the cell. For example, if chloride ions go inside the cell because of activation of some receptor, and we can see this hyperpolarization, so the cell becomes less excitable, and we need stronger signal to induce action potential. So, depolarization, excitatory postsynaptic potential, hyperpolarization, inhibitor postsynaptic potentials. And here in the picture how it works in the whole cell usually. So it's not just one synapse on the one cell. There are a lot of synapses on the same cells, and at the same time there are some excitatory synapses, some inhibitory synapses, and all the effects of these synapses summed and it usually the final integration occurs on the this zone that is called axon helix. And it doesn't show here on the picture, but usually more frequently the excitatory synapses located on the distance from the cell body and inhibitory synapses located near the body. It lets the system to stop the signal transmission, to gate it. So we can, if our inhibitory synapse is activated, then the signal can't transmit from other cells. Then we can just turn off it, and okay, we can see a lot of excitatory potentials, and the action potential is triggered. And we will have now the short overview of different kinds of neurotransmitters and the mainly receptors to neurotransmitters. And is the thing that, again, hopefully you know, but you should always remember that effect of neurotransmitter depends not on the neurotransmitter itself, but on the type of receptors it binds. The same neurotransmitter in different tissues, in different situations, and different stage of life can cause different effects. And you can hear in some popular literature on TV, I don't know, videos, that serotonin is a neurotransmitter of happiness, or oxytocin is a neurotransmitter of empathy. No, there are different receptors, and some of them can cause happiness, if we'll call it so. Another one has quite different effects. Don't oversimplify this system. And we will start from GABA, gamma aminobutyric acid. It is the main inhibitor neurotransmitter in adult human CNS. And there are two general classes of GABA receptors. The first one, GABA A, is ionotropic, and it is chloride channel. So when the GABA binds to these ionotropic receptors, you can see that it's quite complex, a protein complex from several subunits, and these subunits change their conformation, and the channel open. And chloride can go inside the cell, and in adult cell, it causes hyperpolarization and works like inhibitory, has an inhibitory effect. Why only in adult? As I told on the first slide, in embryonic brain, in immature brain, there is high concentration of chloride inside the cell and low concentration outside the cell, oppositely to the adult brain. So when in embryonic brain chloride channels are open, then chloride goes not inside the cell but outside the cell, and it has excitatory effect. But it works like this in embryonic stage. But in adult brain, it is main inhibitory neurotransmitter. And you can also see that ethanol changes the activity of this ion, it increases the activity of this ion, and can cause, that's why ethanol can cause some kind of depression, because it is increased this inhibitory effect of GABA. And the second type of receptor, of GABA receptor, is GABA B. It is metabotropic. It's from the type of G protein coupled receptors. It means that their effect is go through the changes of activity through phosphorylation of G proteins. It's one of very important secondary messengers in the cell. And it modulates calcium and potassium channels, and works and it can cause as presynaptic through autoreceptors and postsynaptic inhibition. And now glutamate. Glutamate is the main excitatory neurotransmitter in human CNS, and there are a lot of different receptors to glutamate. And one big subgroup is ionotropic, and another one's metabotropic. Here on the picture, you can see two kinds of ionotropic receptors, AMPA and NMDA. There is also kainate, but these two types are more abundant. And AMPA receptors is just simple ion channel. It is not selective. All positively charged ions can go through it. Potassium ions outside, sodium ions inside, but because of this concentration gradient, there are more sodium ions go inside the cell, and it totally has excitatory effect. And the second, also ionotropic receptors, is a little bit more tricky. NMDA receptor. At first, it needs not only glutamate to bind to this receptor, but also cofactor glycine. And the second thing, that it's usually blocked by magnesium ion. And this channel is voltage dependent. To remove this ion and to open the channel, the membrane potential need to reach some threshold level. It seems to me that it's about minus 20 millivolt. The membrane potential when the half of this magnesium goes out of the channel, and when this ion is removed and glutamate and glycine are binded, then it also works like positively charged ions channel, and sodium can go inside the cell and calcium, and potassium can go outside the cell. And these two receptors play a really big role in learning processes, in neuroplasticity, and they work together. It works like, firstly, in the membrane, there are a lot of AMPA receptors, and ions go, it works like excitatory ions go, positive ions go inside the cell, and we have this slow depolarization, and in this time, when the membrane reaches this threshold level, then NMDA receptors are also activated. And they can be activated for a long time. So if this magnesium ion is removed, then we have quite a long, relatively long period of time when this channel is active. And this works in the process of facilitation. And the second subgroup is metabotropic receptors. There are also a lot of kinds of different receptors. They're combined in group 1, group 2, and group 3. And group 1 is mainly postsynaptic, and it increases activity of these NMDA receptors. And so they increase the risk of excited toxicity. And these receptors from these groups, group 2 and 3, are mainly presynaptic, and they decrease NMDA receptor activity and risk of excited toxicity. So it's very interesting interplay. It's the same neurotransmitter at the same time activate the receptor, inhibit the receptor, and prevent, increase the risk and excited toxicity and decrease it. And so the system should be perfectly balanced to work like it should. And the next neurotransmitter is glycine. It is inhibitory neurotransmitter in, again, adult human CNS. And it's the main inhibitory neurotransmitter in spinal cord. It works the same way as GABA A receptor, is chloride channel. And the second role of this glycine, as I told on the previous slide, it is cofactor of NMDA receptor. So again, even glycine is at the same time has inhibitor effect through glycine receptor and excitatory effect through NMDA receptor, working as cofactor. Acetylcholine plays a very big role in autonomic nervous system. It is neurotransmitter of preganglionic synapses in sympathetic and parasympathetic neurons, and in postganglionic parasympathetic neurons. It also is a neurotransmitter in neuromuscular junction. Without it, we can't work with our muscles, we can't move. And in CNS, It is mainly found in interneurons. In interneurons. Two classes, nicotinic and muscarinic. And the first one, nicotinic, is ionotropic. It is excitatory because it is sodium channel. And the second one, muscarinic, is metabotropic, and it is G protein coupled again, and can be excitatory or inhibitory. There are different kinds of these receptors. Have several more. Norepinephrine is one of the members of catecholamine family, and it is the main neurotransmitter in sympathetic nervous system in postganglionic neurons. Play really big role in CNS, you know this, and have also as all the receptors are metabotropic, some are excitatory, some are inhibitory in different locations, in different brain areas. We can see a different effect of this neurotransmitter. And dopamine also is the member of catecholamine family, and it plays a big role in reward motivated behavior. It works in motor control. You know that dysfunction of this system of dopaminergic system cause the Parkinson's disease and drug addiction. And there are again 5 types of receptors. The metabotropic G protein coupled, and D1 group is excitatory, D2 group is inhibitory. And you can, and it also works in this negative feedback and so on. And I guess this is the last one we'll talk about is serotonin. It has regulate many autonomic functions, and it's popular, like I have thought, happiness neurotransmitter. But the funny thing, it's the primarily found in the gastrointestinal tract. It's the nervous system that is surround our intestine. But also there's for sure in CNS, for example, in brainstem, and it plays role in mood regulation, inhibition of excessive noise, sleep processes, and so on. And there are 7 types of serotonin receptors. Most of them are metabotropic, but there's also one ionotropic types. They can have inhibitory or excitatory effect. And and why did I tell all of this? So I don't think you will just remember all of this information, all these types of receptors. But again, I think you should at least have this sense of complexity of the system, of these complex interplays at the same neurotransmitters can at the same time, even if the same brain areas have quite different effects. It can inhibit, it can excite, it can modulate in some complex way different processes. And you never should simplify this history. It just don't work just like this one is excitatory, this one inhibitory. No. And and also remember that there are a lot of different synapses, different receptors on the cell, and what we can see totally, and what we can see on EEG and in other neurovisualization methods is the sum of all these processes together. And the next slide, we will have another quiz. It will be, it seems to me, 6 questions and 5 minutes. So let's start, if there's no questions. Okay, let's start.
Do we supposed to see the questions on the screen?
If there is a question, sorry.
Yeah.
Погромче можете включить микрофон?
I was just confused because the slide hasn't changed, but that's okay now.
Yeah, okay, yeah, sorry, it's my fault. Send your answers, please. And yeah, let's see. Let's start from the beginning. Yeah, ionotropic, negative, positive, glutamate kappa. Yeah, great. Chronic. Yeah. Mm. Yeah, here a lot. These questions I meant not what kinds of ions, but the charge of the ions. Yeah, but I I see. But I meant negatively or positively charged. Mm. Okay, Daniel, you can find this information in the presentation or somewhere else. Not a problem. Yeah, great. Yeah, really great. And we have just one more quiz later, but then it will be done. So move on. Yeah, and now we are closer to EEG, and we will talk a little about brain source of MEG and MEG, as was called the lecture. And first of all, the EEG and MEG really very close methodologies. Maybe Alex will then tell why not, but from physiological point of view, it's very close. And in first of EEG, we're measuring electrical current, and in the case of MEG, magnetic fields, but both of them are reflect some postsynaptic potentials and also non-synaptic intracellular events. We will not talk today, at least we will talk a lot about non-synaptic intracellular events, and there is, but they also contribute to the signal we can see with our methods. And there are subthreshold oscillations, some calcium spikes, glial signals. So now it is known that really glial signals play a big role in all the processes in the brain. They're not just supporting cells. But we will talk mostly about postsynaptic potentials now. But if you are interested in the topic, you can also then read more about these non-synaptic events. Why do we measure postsynaptic potentials, not presynaptic? Let's compare. Here we can see this action potential or presynaptic, and here postsynaptic. The action potential is very short, it's about 2 milliseconds, and it is biphasic. To see some signal on EEG, for example, we need about at least 100 of cells excited simultaneously. And we understand they really don't excite absolutely simultaneously. In case of this biphasic signal, if there are a lot of cells and some of them activated here and some in maybe here, we sum all the signals and we just get 0. We can't see the signal because of these two phases, positive and negative. And a postsynaptic potential is monophasic and is quite long-lived. So even if cells don't excite at the same time or don't have these postsynaptic potentials at the same time, we can summarize it, and we can see the EEG or MEG. So I have some problems with the internet. It's nothing. And the main contribution makes these postsynaptic potentials from dendrites of the big pyramidal cells. This is special kinds of cells in our brain, and they have big dendrites that are oriented in parallel and perpendicular to the cortex. So all these signals, if these cells have work at the same time, all these currents go in, and we can see all currents, electric currents or magnetic that's produced by all these cells simultaneously. We need at least 100 of cortical neurons on some not very big area you see to detect this signal. And here at the bottom is super nice picture. I like it very much. Just these stained pyramidal neurons with long dendrites. You can see how it looks in the real. Yeah.
And Masha,
Yeah.
I'm sorry, I have a question of the previous picture. On the bottom one, so how many millimeters or whatever micrometers are shown, like the raster from left to right? Your guess.
From from left to right.
Yes. Well, I mean the width of this picture in physically, how large it is.
I think we can compare with this scheme, as here we can see this the height of this neuron is about 2.5 millimeter. So in this neuron, yeah, so we can compare with it. So it looks from left to right.
So it's actually not that small. Yeah.
Not
Okay.
small, yeah, so they big. They called big.
Yeah. Okay, cool. Yeah. Thank you.
Chat something of the upper two pictures. You mean, sorry, the previous slide. Okay.
Yeah, the previous slide. Sound was not good, and the part with perpendicularity.
Yeah, I tried.
I didn't get the part.
Okay. What I talked about, that these dendrites are parallel to each other. Yeah. So they go in the same direction, we can see. And they located perpendicular to the cortex surface. So here's the surface, yeah, the cortex, the skull, for example, and they're perpendicular to it. No, yeah, you can ask me later if something unclear. Yeah, and these postsynaptic potentials induce dipole. It's quite a complex part for me as I'm not physics, but I will try to explain how it works and how I understand it, and probably some Alex, somebody say if it's wrong. So we have the synapse. For example, here in the picture, it is excitatory synapse, and it causes influx of positively charged ions inside the cells here on the synapse. And here we can see inside the cell, we can see increase of positive charge. And on the distance from this place, there is relatively negative charge. So here is plus and here is minus. So the primary current, the current intracellular current in this dendrite is going from top to down, from plus to minus, this direction. And there is also extracellular currents, secondary currents, that goes opposite direction. As here is minus, because a lot of ions, positively charged ions, go inside the cells. And here is a relative plus, relative to this part. So here, outside the cell, this current go opposite direction from top, from bottom to top, up, down, oh, yeah, down, up, sorry, from plus to minus. And this extracellular currents is what we can detect with EEG methods. Is it clear? Or we need to say it one more time? Okay, so excited. We will have quiz on this picture.
One more time, please.
Okay, yeah. We will have later quiz on this picture, so let one more time say this. Excitatory synapse. It causes influx of positive ions inside the cells. And here inside the cell, we have plus. On the distance from this place, from this synapse inside the cell, we have minus. Because here are a lot of positive ions, here is the same amount of positive ions that were previously. Yeah, is it clear? Okay, plus, minus, outside the cell. The opposite situation. Here is minus. Yeah, a lot of positively charged ions go inside the cell. So here outside is minus. And here is plus. We're talking about relative charge, actually. So here is more plus than this place. Yeah, so here from plus to minus, and current, you remember from physics in school, goes from plus to minus. So inside it goes this direction, and outside it goes opposite direction. Always from plus to minus. Yeah, and with EEG, we detect this secondary current, this extracellular current. And with imaging, we detect the magnetic field that is produced by intracellular current. Yeah, and here on the picture, this on the left, there is mistake. When I prepared the lecture this time, I understood that it's not really correct. And the first my idea was to remove this picture, but then I decided to ask you, what's wrong with this picture? Somebody? Ideas? Sorry. Sorry.
Please, can you repeat the question?
What's wrong with this picture? I will then go to this chart. Based on what I taught on previous slides.
Maybe it's an axon, but not a dendrite?
Yeah, it's true. On this picture, I found it somewhere in the internet, but it looks like we register the signal from axon. No, it doesn't work like this. It really should be this long dendrite here on the upper side of the cell. So it's not very correct picture, but the idea is correct, but not the picture of this cell.
Yes, beware of what you can find on the internet, especially with open chat AI. Yeah.
Okay, it was something. Yeah, the cell membrane is the ions can move inside. Yeah, the question in the chart. The ions can move inside the cell. So if it's immediately distributed over the whole cell, then yeah, it will be just the charge go inside the cell and the whole thing become more positive. But really, no, that have some electrical properties. Yeah, so there are some parts of the membrane that is more positively charged and another parts that are less positively charged or negatively. So it's if it will be just круглая лошадь в вакууме, so some very theoretical system, it could work like just the empty space and the charge go inside this empty space. But no, we have the living cell with the cytoplasm with some processes inside of it. Nothing happens immediately, and there are not равномерно. Okay, I forgot the word.
Uniformly, maybe.
yeah, not uniform distributed inside the cells, all the ions and all the substances.
Okay, thank you.
Yeah, and the next, it was again when I prepared this lecture a couple of years ago. It was really new for me idea. It's about EEG and what means the signal of EEG. We can see what it means in terms of the origin of this signal. Here on the picture, on the left, we can see two situations when on EEG we can see some positive wave. And on the left, on the right, there are two situations when we see negative waves. But if we look what happens inside the cell in this time, there are two quite different processes. Here on the left, we can see superficial excitatory input. So as we talked about excitatory input, plus here inside the cell, minus here, extracellular opposite side, plus, minus, and we can see the current then go this direction. Bottom up, yeah. And we can register it and see this positive wave. And here, left, right, there's deep inhibitory input. So here's minus inside the cell, plus inside the cell, and in terms of currents, of extracellular currents, it is the same current, and we also see the positive wave. But actually, from this picture, from this signal, we don't know if there is some excitatory signals, other some inhibitory inputs. Now we can see it in the same way. And on this right picture, also the same situations, but here is deep excitatory input and superficial inhibitory input. So there are big questions to information about some complex brain processes that we can get from EEG. Alex will then explain how we can solve it, but from physiological point of view, it's a very questionable thing. And even if we can see some strong positive effect on EEG, theoretically, it could be summation of some amount of excitatory inputs and some amount of inhibitory inputs, or it can be a lot of inhibitory inputs or a lot of superficial excitatory inputs. You never know. Yeah, and about what signals we can see with EEG and imaging. With EEG, we can see dipoles that are located tangential and radial dipoles. So in both cases, there is here below there are secondary currents, and they go in some way they go out of the skull, and we can see them with our electrodes. But in case of MEG, we can see only tangential dipoles. Why is it so? Because if there is some radial dipole, the magnetic field that is go around this primary current will go in parallel with the skull here on the picture, right on the bottom. Yeah, so it will just go in parallel with the skull inside the brain, inside the head, and we can't detect the signal with MEG. But with EEG, there's another problem. As we measure the relative signal, we measure the difference between our recording signal on our recording electrode and our reference electrode. It depends on position of these electrodes. And here you can see that, for example, if we put reference here for longer distance, we will see the higher signal than if we will put the reference here on the closer distance to the recording electrode. And even more interesting, if our dipole is somewhere here, in between of recording and reference electrodes, we just will not see anything. because there will be the same signal on the recording and reference electrode. It's about limitations of the method and how we can interpret it. Yeah, so to summarize, EEG, we measure relative signal. It is also affected by changes in conductivity. Usually has quite long preparation time, especially if we are using wet electrodes and you should to place gel. Now there are also some dry electrodes and probably some of them works quite good, but still the most of clinical EEGs, scientific devices use wet electrodes with gel. So it makes the procedure more difficult. There are some difficulties with modeling of the sources, Alex will tell, and lower spatial revolution. But what is good, the head movements are not disturb the signal too much, so we can work with it. And in case of MEG, we measure an absolute signal, and it's not affected by differences in conductivity. Probably has shorter preparation time, but you should go to this MEG center. It's quite far from HSE, has higher spatial resolution, but very sensitive to the movement of the head. You will have really bad signal if the head is moved in the camera. And probably the main problem with the MEG, it's a very expensive and bulky acquisition system. And just to compare, here on the left, there are some nice pictures of portable EEG systems, some of them even not too expensive, and probably you can buy it yourself and use at home. And a MEG system is very expensive and big, and it's not for home use for sure. And then we will have 3-minute quiz. It will be about currents, as I thought. So just last try. I believe in you. Yeah, let's start. 3 minutes. Okay, please send your answers. Okay, so here not all answers are correct. Let's see. Okay, let's look in the picture. Here it is inhibitory postsynaptic potential. As we talked previously, and you answered to this question, inhibitory postsynaptic potentials, it is influx of negatively charged ions inside the cell. So here will be minus. And then starting from this, minus, plus, minus, plus. Yeah. Is it clear? So we just need to understand this first thing. Here will be minus because of the inhibitory postsynaptic potential. Yeah. And then we can just calculate what will be in the other points. Everything right? You mean your picture? I can't find your answer now, but yes, you need -1 is minus, 2 is plus, 3 is minus, and 4 is plus, because here is the influx of negatively charged ions. Okay?
Understood.
Yeah. And the last part of our lecture, it will be about the origin of EEG rhythms. And actually, I had quite a lot of slides more, but this time I decided to not tell you everything I have here on the slides. I didn't remove them. You will have this presentation, and if you like, you can read it yourself, but I decided it makes the lecture too overloaded, and I will tell about some general principles. And first of all, just to remind you the organization of the cortex, of the brain cortex. Then our cortex has several layers with different neurons. There are granular cortex when we have the full set of the layers, six layers. There is agranular cortex, where there's about 4 layers and something intermediate, this granular cortex, where we have these small 4 layers, so it's something intermediate. And of course, different layers contain different kinds of cells with different functions. And what is important is there are ascending and descending pathways. So ascending pathways is pathways from subcortical structures, from thalamus, for example, to the cortex. And the main target of these ascending pathways is this layer 4 that is absent in this agranular cortex. This is granular layer, and its main function is to get information from thalamus. In this agranular cortex, there are also some ascending pathways from thalamus, and they are going to in other layers, but are much more weaker. And it's explainable as granular cortex is mostly located in sensory cortex, so we need to get a lot of sensory information from thalamus. And agranular cortex is mostly located in the frontal lobe, it's motor cortex. It just don't need a lot of information from thalamus. And there are also descending pathways. The cortex sent information down to the subcortical structures, to the thalamus, to the basal ganglia, and so on. And this interplay between these different structures, between cortex and subcortical structures, play really big role in all these EEG rhythms, in these rhythmical oscillations. I'm not sure this picture should be here, but let's explain it here. It's one of the nice pictures that depicted this interplay, and it's again about negative feedback, about feedback system we discussed when we talked about autoreceptors. You can see here this schematic picture how neurons in different cortical layers can interact between each other. Actually, there is the same situation with subcortical structures. Here with blue triangles depicted excitatory neurons, and with red rounds, red circles, inhibitory neurons. And what happens here? This excitatory neuron, let's start from this, send excitatory inputs to this and this neuron, to this excitatory and to this inhibitory. This excitatory neuron sends back the excitatory input to this cell. So here it evokes positive feedback. But at the same time, with this cell, this inhibitory cell is excited. And this inhibitory cell inhibit this neuron and also inhibit itself. And here works negative feedback. So what result we can see from bottom of this system? We will not even talk about this. Let's see only this, look on these three neurons. For example, these neurons start excitation. It also excite these cells, and we can see this some signal, for example, some excitation, some excitative potential on EEG or somewhere else. But after that, when this cell is activated, it stops the signal transmission. And we can see the next path, so we can see inhibition of the cells. But at the same time, this cell inhibit itself and stop working. And now this cell can send the signal again, and we can see the next phase, the next oscillation. And we will talk about it one more time, but it's just the main principle, how it evolves.
I have a
Yeah.
question, Masha. Masha, can you go
Yeah.
back? Yes,
Yeah,
I have
yeah.
a question. This arrow that goes from the bottommost excitatory neuron to the topmost inhibitory neuron, and the reaction of this inhibitory neuron, is it by definition slower, so that the inhibitory effect induced on the top excitatory neuron is sort of delayed, right, with respect to the excitatory effect that is used, that is affected, that the top neuron is affected by from the bottom neuron. So is it this blue red blue loop?
Mhm.
It's got to be slower, right? It got to take longer time for the excitation for the
Probably yes. It's very approximate scheme. It's it can be it's definitely it's not one neuron. It's a more complex system. Yeah, it can work. It can work not absolutely simultaneous. You can need not only one excitatory input here to excite the cell, or maybe several excitatory inputs. Yeah, but to make the system working, yeah, you need some time delay.
Yeah, and another question, I think what you said that once the top neuron gets excited, you said that we can observe the excitation by action potential, but I think what we actually observe is the fact that the bottom neuron is exciting, is transmitting the signal to the dendrites of the top neuron, and this is this propagation down the dendrites of the postsynaptic potential between the bottom and the top neurons, right? This is what we see, right? So
yes, on EEG we will see the signal that goes along these dendrites, so not the excitation of the cell itself, but the signal that is going.
But at the same time, we will also see the effect of inhibitory neuron on the dendrite, right, of this top neuron, right? We will also see it in EEG and MEG, and in the MEG, actually, it turns out we will see it in the same direction because the excitatory synapses are further from the soma, and the inhibitory synapses are closer to the soma. But for the excitatory synapses, we have negative charges, right? And for the inhibitory, we have positive charges, or whatever, vice versa, whatever. So, but yeah, but they will create the current in the same direction. That's yeah.
Yeah,
So
if we see, yeah, so if we see the signal that is going to both of these neurons, it will be the same, definitely. And is it excitatory signal to both of these neurons? And okay,
yeah,
okay, yeah, so
it's just but basically to show that that in fact, although it's kind of there is a legend, what we see in EEG and MEG, it has to be still taken as a legend in a way. So it's like zero level approximation that we all rely upon.
yeah, so
But
it's
but
explanation how it can work, that we can see some oscillations. So why there is some, why there is signal, there is no signal, or there is one direction signal.
Yeah, so it's important that to see the oscillation, you have to have the excitatory part, you have to have the inhibitory part, and there has to be a delay between inhibition and excitation. So, and the excitation itself has to depend on the level of activity, and then they kind of ping pong between these two states.
Yeah, so it's interplay between different kinds of neurons.
Yeah. Okay, thank you.
Yeah, and now this EEG and MEG rhythms, guess not new information that usually distinguish several frequency bands. Gamma waves are the most frequent from 30 hertz and upper, and the delta waves are the slowest ones. And there is quite a big area of science that studies this behavioral and physiological correlates of different frequency bands in different brain locations. And yeah, we will not discuss it. You talked about it on other courses. But again, I'm skeptically always. So we'll talk about limitations for EEG rhythm science. And the first limitation is these frequency bands are artificially defined. So at first it was absolutely artificially defined, then some more complex mathematical methods appeared, and you can see the spectra, and you can say that, okay, let's say that this one is some special rhythm. And there are some correlates and reasons why we think that these particular frequency bands really have sense, but still it's also approximation. There is no some special structure that produce this rhythm in precisely between I don't know, 8 and 12 hertz. No, it doesn't work like this. Then about all studies and all information we have about rhythms and about neurophysiological basics of these rhythms, the most of cellular recordings that can explain the cellular processes underlying these EEG rhythms made in animals. And of course, we are quite similar to at least some animals, but never can be sure that really in human brain in all situations will work in the same way. Also, if we're talking about some intrinsic properties of the cells, the most of studies were done in vitro or in cell cultures or in slices, and it gives a lot of information. It's super important and interesting studies, but also we can't be sure that in the whole brain the system works in the precisely same way. It can work. There's much more connections in the whole brain, much more extracellular space and glia and so on. So now you can use some co-culturing procedures when you have not only neurons in your cell dish, but also glial cells or maybe even some other cells, but it's still the model, not the whole brain. And if we're talking about study of the network interactions, they made their conclusions made from recording from specially discrete locations. And we can make the assumption that we can approximate the results we have from these special discrete locations. And we can think that it means that somewhere in between these locations, we will see the effect and we can make modeling. But again, it's approximation. And the last state is from article by Cohen, the ultimate but ultimately unattainable goal, one-to-one mapping between EEG feature and microcircuit configuration. It is the same what we have talked on the previous slides, that looking on EEG, we can't say what really happens in the brain in this moment. And about generation of different kinds of rhythms. I highly recommend you this book. I send it to Daria, but if needed, I can send the whole book in PDF one more time. And there is special chapter that reviews different studies in this brain rhythm generation. And I will just say, discuss several theories of brain rhythms generations and tell the main logic of these theories. Here's on the picture 3 ways. The first one is pacemaker, second is intrinsic, and the last one is synaptic. For my opinion, these first two can be, it's the same. And this means, this pacemaker or intrinsic theory means that there is some source that can just rhythmically generate, for example, action potential or some rhythmical activity. But in the first case, here on the picture, pacemaker means that there is some extracortical source, for example, somewhere in thalamus. Intrinsic means that there is such pacemaker inside the cortex. But the principle is the same, just some rhythmically oscillating cells. And synaptic feedback theory, it is what we have talked about, that there is some system, some network that inside of which there is some interplay between inhibitory and excitatory neurons, and we can see these oscillations because of interplay of these neurons. And if we're talking about this pacemaker theory and pacemakers, here's just one example that was studied, it seems to me, in cats, but not sure. This thalamic delta oscillation. It was found that there are different sources of delta rhythms, and one of them is found in thalamus. And then it was found that there are special cells in thalamus that can produce this oscillation. Why it happens so, how it works. In these cells, there are special low threshold calcium channels and also hyperpolarization activated cation channels. So in the normal cell, remember there is resting potential about -70, and when the cell is hyperpolarized, -90 millivolt, it is less excitable. But in this cell, when cell is hyperpolarized, for example, -90, that now the precise value, then this hyperpolarization cation channels are open, and we can see slow depolarization. And then when it reaches the threshold, the next channels, low threshold calcium currents, are open. And we can see there is influx of calcium ions, and then start action potential, and we can see this burst, this acute depolarization. After that, like after every action potential, we can see repolarization. But in normal neuron, in Evert's neuron, after hyperpolarization, we will see the coming to the resting membrane potential. But in this case, on this hyperpolarization stage, these hyperpolarization activated channels are open, and it's go again. And if nothing blocks this system, it can do it, it can oscillate all the time, just itself without any external signals. And why it doesn't work, why we can't see this thalamic delta oscillation all the time, is because of the cortical descendant pathways that just inhibit these cells in some cases and in some not. Yeah, so this is pacemakers, and these kind of pacemakers can be in thalamus, can be in some cortical cells inside the cortex. and it can cause some of the rhythmic oscillations. Yeah, and this is actually the same as we discussed previously, this network situation, network hypothesis, when there is some excitatory neuron and inhibitory neuron. And for example, here on the picture, there is some external input that activate this excitatory neuron, and that it goes like we discussed previously. It's activated excitatory and inhibitory neuron. Here it's in the same level. And then in some period after excitation, this inhibitory neuron block this cell or this structure. And we can see, for example, repolarization or whatever. We can, as the main thing, we can see these rhythmical oscillations. Yeah, it was the main I wanted to talk to you today. Again, two main theories of rhythms generation. Actually, both of them are working in our brain. It's the interplay between this interaction between these two kinds of generations. And when you're studying any EEG rhythms, you should remember that it's artificially frequency bands, and even in each frequency bands, there are different kinds of these rhythms. Some of them, for example, for data, one of data rhythm is found in cortex, another one is produced in thalamus. It is the same for another rhythms. They have different sources. Probably sometimes you can find the primary source of this EEG rhythm, sometimes you can't do that. And just looking on the EEG and on the power spectrum power, you can't say where these rhythms come from. Yeah, these slides you will have, but I will not tell about them. And just to summarize what we have talked today about, first we started with this familiar for you things about resting membrane potential and action potential, then talked about signal transmission and receptors and different kinds of receptors, and then talked about postsynaptic potentials and what signal we can detect with EEG and MEG, and this maybe more complex dipole story, and just several words about brain rhythms generation in our brain. Thank you. If there are some questions, you're welcome.
Any questions?
If not, then Alex, continue.
Yes, I will then. Okay, Masha, thank you very much. I want to tell you that what Masha has managed to compress into like 1.5 hour her lecture, or maybe a bit more, is a material of, I would say, 2 or 3 classes, so that you don't have to feel overwhelmed. But some of these classes you are going through in the program, and this class today is not, this class, I mean, this mathematical aspects is not about all these detailed neurons and excitatory inhibitory potentials. We will be operating on much more abstract level, although Masha mentioned that these things should not be simplified. Well, but we are simplifying them, and actually not we are simplifying them, but the methodology that we are using, the one that is based on EEG and MEG, is the macro scale methodology that allows to register macro scale events. And this is like, you can imagine that if you fly over Earth in an airplane and that you see all these mountains, right, and all the river floating, so you're of course not caring about movements of individual sand particles in the bottom of the river, right? So but you're looking for the general flow of the river. That's what we are going to be able to do and afford with EEG and MEG. Okay, so Dasha, are you there? Are you ready?
Yes, I'm ready. Do you hear me?
Okay, so now according to the plan, because I want to postpone my participation as far as possible, so I asked Daria to make a small, very brief introduction about the program to software tools that we are going to be using in the course, and compared to the last year class and one before the last year, I think we will try to make it a bit more practical this semester, so that we can give you some hands-on experience on analysis of some real EEG data in different paradigms, starting from simple evoked potentials and then go into even brain-computer interfaces, so that you, by the end of the class, you have some hands-on experiences dealing with the data. And to do this, you will need to cope with Python, however ugly and horrible it is, but you still have to, because this is the default standard now. And it's like with latex, right? Latex is not great, but there's nothing better. So yes, Daria, please.
Yes, thank you. So I'm glad to see everyone today. And actually, I will see you in person on Friday, where I will explain all the details related to the introduction to the software which we will use to process EEG and imaging data. And now I will just cover some things that you need in order to install Python if you don't have it installed, and just explain some basic stuff. And I expect that not all of you have experience in programming per se. Of course, as far as I know, at HEC, there are some introductory programming courses you should have had in autumn, but okay, I will try to adapt to your level, and we will see how it goes. So as Alexey Evgenich said, you will need to use Python, and the simplest way to download it is to use the Anaconda distribution. Actually, I will send you the script with information about, with all the links I will provide now. I just don't have the script right now because our server has died, and so I will send you it later today. So you need Anaconda. Anaconda is a package manager and a Python distribution. You can download it for any operational system you have. It takes some time. And when it finishes and you install it, you can actually run the special environment which will allow you to organize your scripts and the code. So let's wait for it. So by default, you already have the needed environment. I recommend you to use JupyterLab. I know that, for example, Alexey Evgenievich doesn't appreciate this environment because it's much more concise to organize your scripts in just as scripts. But for the sake of processing EEG data and EEG or imaging data, with making the beautiful visualization, making some brief reports, maybe it's a good idea to use the format of so-called Jupyter notebooks. What is Jupyter?
Yeah. No, I'm not against Jupyter notebooks. Yes. No, I'm just saying that it is very well, unless if you're using it for routine development, it becomes very chaotic. But it's a great tool to present the material, to present the stuff. Basically, once you have things working, to put it in a notebook, to share with colleagues, to explain to students, it's a great tool because on the fly you can change things and you can recompute. But if you use it routinely, I mean, for development, it's very easy to not change, not recompute something, but recompute something else, things like that. So,
a good principle of programming culture and I suppose that I will cover some of these principles later, because it becomes very important when you have a lot of data, a lot of projects, and a lot of functions. A good principle is to put your functions, which may be very long, into separate scripts, and address them from your Jupyter notebook, and make it brief and concise in terms of visualization. But this was just a small nuance. So in these notebooks, what can you have? You can have the main element of these notebooks is the cell. The cell may contain any code, or the text, which is organized using so-called markdown. So you can write any header, any text, and just see it in a beautiful way. And later you can just put various code in your cells and run them and see the results. So let me create a clear notebook. And the Python, as any programming language, allows you to perform simple operations like simple computations, or putting into the output some strings. So here, if you have some intuition about how the programming works, there won't be any problems. In some cases, Python can be very tricky, because it can pass away some bugs and so on. But so you should be careful. You should be more careful than, for example, with MATLAB or C++. But you will get used to it. Yes, and of course, our course is not about programming per se. So if you feel that you need to cover some basic principles in programming, I recommend you to use so-called data camp resource, where you can learn the basic principles in the real, I could say, environment. So let me show you how it usually appears. Oh, it wasn't supposed to happen, sorry. Give me a few seconds. So here in this data camp, you can choose small courses which are available for free. For example, you can choose Introduction to Python, and just listen to various tutorials and perform some exercises in the in this environment, which will demonstrate you the correctness of your script and the results. It will take several hours, and maybe it will help you to cover this intuition for our future purposes. But if you don't have experience, actually, it's not a big problem when you use the software I will describe you later, because actually it is supported by good documentation, and if you just understand what a piece of the script does and know how to change the parameters, you don't need to write the script from scratch. Yes, what software will we use? Sorry, I see. Okay, I saw just a few messages about having a break, but I suppose that I will be very brief, and you can have a break later. So we will use the software called MEG Python. Here you can find the website with documentation. In order to install MEG Python, you should go to your terminal, and write the following command, which will install this package.
May I interrupt
Yes,
for a second,
yes,
please?
yes.
So it's in general, well, with all this mess that Python is causing, it's much better to install M&E Python into a special environment. So
Oh.
you can create Conda environment, call it like M&E, whatever, and then you install everything into this environment, because it will require some extra packages and versions of packages that may not necessarily be the best for the rest of your works, right, during other classes or just your own coding, so that it's better to create M&E environment, just conda n of whatever minus name, whatever, create something like this, and then activate this environment and do what Dasha says to you within this environment. So then in brackets on the leftmost side, you will see not base, but you will see, let's say, M&E.
That's the name of the environment. Yes,
Yeah.
thank you for this commentary. Actually, it's a good idea in general to create environments. I will put in the script that I will send later the instruction how to do it and what it means. But actually, for the maybe 1 year or 2 years, I didn't have any problems with compatibility of MEG Python and the packages it requires, even if I don't use the environment. But yes, actually, it's a good practice. So when you have MEG Python installed, you can import it to your script. Yes, and this is one of the specific properties of Python, that you need to import the packages. Sometimes you need to import the packages from outside. Yes. Now you have all the needed instrumentary available for you. I recommend you to just to look at the MEG Python documentation before Friday in order to understand what you can expect from it. Actually, there is a very useful page in this documentation called the typical MEG and EEG workflow, which demonstrates you what kind of data you will have to use and to process. So what we will have? We will have the raw data. This is the time series of your EEG or MEG or invasive recordings, or just the time series you need to use from other sources. On the other hand, there is a structural information, because usually we have to use MRI information about the structure, anatomical properties of our brain. And I will show you in details how to segment this structural information in order to make it applicable to all the usual pipelines in MEG Python, and actually not only in MEG Python, but in Brainstorm. But you use Brainstorm in MATLAB, and now MATLAB has restricted its licenses, so I don't think it will be a good idea to cover it in details during this course. Yes, and then you can process your raw data in different ways and combine it with your structural information, and finally get very useful entities from this information, such as averaged response to your stimuli, for example, or forward solution. Alexey Evgenich will cover in details what does forward solution mean in mathematical terms, and I will demonstrate how to create forward solution and how to compute inverse operator from it in practice, and what details of preprocessing can affect your results when you need to estimate the activity of the cortical sources in your cortex, because actually some of the techniques of forward and inverse modeling are very sensitive to the parameters that can seem unnecessary for you, for example, noise covariance matrices and so on. So I will show you different examples of good and bad inverse modeling, and you will understand how to find the balanced solution. Yes, so it's a good idea to walk through this website and see all these beautiful tutorials, which are usually updated. Yes, and And starting from the Friday, we will start to work with actual examples. And I hope that you will be able to not only to repeat after myself, but after me, but to create your own solution. For example, we will think about it, but for example, you could decode the motor imagery from EEG data or localize sources of some typical evoked responses from the real data. So I think that that's it. And until Friday, I need you to do the following things. I need you to install Anaconda, to check that you can open Jupyter Lab environment, that it works, and I need you to install MEG Python and check that it's successfully imported. If you have any problems, you can contact me directly until Friday. I will send you my email and my Telegram contact, but I don't expect any technical problems here. So that was my organizational part. Thank you. I suppose that Alexey Evgenievich has left, but I know that he planned to give you a few points. Alexey Evgenievich. Yes, you have your microphone turned off.
Yes, hi. Sorry, I just went to grab tea, a cup of tea. Okay, so yes, Dasha, thank you very much. So Dasha has just given you a very short
sorry,
and brief.
Alexey, I see a question in the chat. Irina,
Yes, please.
You ask if you can use Google Colab. I don't recommend it for the following reasons. First of all, Google Colab, there is a limit for its working. So if you need to compute long scripts, it doesn't allow you to reach the time over several hours, and it's actually a problem. The second problem is that it doesn't allow for interactive mode, and when we work with neuroimaging data, we need to scroll the time series, we need to check the orientation of different dipoles, for example, and we need to actually use the plots in interactive regime. That's why it's a good idea to use MEG Python locally. So you should account for it. So
yeah,
that's all.
Any more questions? Guys, if you are those who speak Russian, I mean, so if you for some reason you are shy to ask questions in English, I don't know, I mean, maybe you can either type them in or you can just simply ask them in Russian, and then I will translate, or Dasha, or we will translate them to the rest of the class so that you can, freely communicate. Okay, so all right, so Dasha gave you a brief introduction of like software tools that were actually very simple set of tools: Python, the MEG Python, and the necessary Conda environment, which Conda management software that allows you to somehow get control over the multitude of Python packages. Okay, so I will now give you, so again, we are going without much of the break. So if you want me to give you a break for like 10 minutes or something, I will actually be more than happy, but it's up to you. So if you decide to give that we'll make a short break now, then let's do so. Otherwise I will continue. So what will you?
Yes, please, let's have a short break.
Yes, okay, yes, do some exercises, things like that. Yeah, so for 10 minutes. Okay, so we will meet in 13:25, okay, Moscow time, and I will stop, pause the recording now. Okay.
Okay, thank you.
Thank you.